start to have tails, and these tails can be chemically modified by methylation, acetylation, ubiquitination, simulation, and so on. And these modifications associated with different transcriptional states, so chromatin states. For example, if you see trimethylation on lysine 4 of histone 3, you're almost certain that you're in promote region of a gene which is either active or can be activated soon. And if, for example, in addition you have trimethylation of lysine 27 on histone 3, you are almost sure that this gene is repressed, and you know the mechanism of repression, so you know that the gene is repressed by polycomb repressive complex 2. And if you have two marks together, you know you're in the region of repressed promoter. So today I'm going to focus on another mark, which is acetylation of lysine 27 of histone H3. And this is a mark of active promoters and enhancers. It looks like this. So here I'm showing you a screenshot. This is a gene, transcription start site of this gene, and this is distribution of this K27 acetylation mark obtained by ChIP-seq experiment. And below in blue you see a track for this promoter mark, K4 trimethylation I just mentioned. So you see that even if you don't have expression for this gene, as you see these two marks here, promoter mark and active promoter or enhancer mark, you know that this gene is active and it's transcribed from this region. Here on the left you see another region where you have some enrichment of this K27 acetylation mark, and you don't see any promoter mark here, so you can think that this region is an enhancer region. And you see that the signal is quite narrow, so here it's only 3 kb. And quite some time, Rick Young and James Bertinoro, they discovered or named another type of enhancers that they called super enhancers, which are much wider regions, having lots of K27 acetylation and having very strong regulatory potential. So here I'm showing to you an example of this super enhancer, which is 240 kb long. So 3 kb here versus 240 kb here. And so you see lots of acetylation, and you can think that here you have many individual enhancers, like here and there and there and there, but also you have a lot of acetylation mark in between these regions, and this is the way to find the super enhancers. And it turns out that genes which are regulated by the super enhancers will be highly expressed, and the more acetylation you have in this region, the stronger will be the expression of these genes. So here I'm showing to you an example of the correlation between super enhancer score, which is measured by this just density, some of the density of this profile here in log scale, versus gene expression, again in log scale, and this is for GATA2 super enhancer and neuroblastoma. And we see the correlation coefficient is very strong, so each point is a neuroblastoma cell line in orange or normal neural crest cells in blue. So the more acetylation you have, the stronger is the super enhancer. And the common way to identify the super enhancers is to first find candidate regions that have a lot of marks. And the way to do it is to do peak calling. Peak calling, it's a part of ChIP-seq analysis. And here what you do, you find regions of enrichment, you call it signal, and this you call background. Then you fuse these regions together using a distance of 12.5 kb, which is, I think nobody really knows why people use it. And there are efforts to find the best distance to fuse these enhancers to 5 super enhancers, but in my hands they don't work. So when you fuse these regions into bigger candidate regions, then you can rank them according to the number of K27 acetylation reads, and then you arbitrarily cut it here, and you say these regions are super enhancers, and you have few super enhancer regions with lots of K27 acetylation signal, and all other regions you call them enhancers, and they have much less K27 acetylation signal. The method to do this super enhancer calling is called ROSE, and it was proposed by Rick Young and James Brodner for normal cells, and we modified it in a method which is called Lily, and I'm going to talk about it a little bit later. So why these super enhancers are interesting? First of all, because they can point to very important transcriptional regulators that determine cell identity. So if, for example, you take a gene which is expressed in different tissue, it's likely to have different typical enhancers in different tissues. But if you take a gene, especially a gene coding for a transcription factor, which should be expressed in one particular tissue, and the expression should be high, and the product of this gene will determine cell identity for this tissue, then it's likely to have a super enhancer. And in addition to K27 acetylation mark that I mentioned that you can find in these super enhancers, you can also see lots of RNA pol2 binding on the super enhancer regions, as is shown here. Acetyltransferases, BRD proteins, HDACs, and lots of different transcription factors will bind the super enhancer regions, compared to typical enhancers where you see much less of such binding. In cancer, in this publication, they propose that de novo super enhancers can be formed or activated, and that these super enhancers, they turn on genes with oncogenic potential. So they marked super enhancer in this colorated cancer cell line, and they associated genes to the super enhancers, and they show that genes that the strongest super enhancers correspond to invasion metastasis. Then second one, it's sustaining proliferative signaling. Third one, inducing angiogenesis. Then tumor promoting inflammation, and so on. So it looks like cancer finds a way to turn on genes it needs to invade, to proliferate by creating de novo super enhancers. However, I don't know much about how the super enhancers can be created. Maybe you have some ideas how they can be created or activated? No hypothesis? Rearrangements. So yes, so there's a couple of papers where they show that super enhancers can be hijacked. For example, for the third gene, which is very important for cancer, you can create a rearrangement and hijack a super enhancer or enhancer, but it's not a way to create it. It's a way to bring it closer. Maybe, but how do you change it? So, yeah, actually it's an interesting hypothesis. I think nobody looked at it. So, yeah, you can somehow maybe methylate CTCF binding sites and change the 3D structure of chromatin. We need to explore it. Thank you, guys. So, I know about 2 examples of a genetic event where a super enhancer can be created by a small mutation. So this is a published one. It's a non-coding mutation upstream of the TAL1 oncogene in T-cell acute lymphoblastic leukemia, and this small modification creates a de novo binding site for the MYB transcription factor, and for some reason, just the creation of one binding site for MYB can create the whole super enhancer landscape around it. And another example was given by Bark de Plancker recently, but he didn't publish it, so I'm not going to go into it. So my hypothesis, in addition to Eva's hypothesis of rearrangements of 3D structure, is that you have a general rewiring of transcription factor networks due to copy number aberrations or small non-coding modifications, maybe coding, that results in changes in the expression of transcription factors, changes of the regulation, and it can create, activate de novo some regions in the genome. So, and there is a very useful concept to study this rewiring of transcription networks, which was again introduced by Rick Young and James Brodner, which is called co-regulatory circuitries. Have you heard about co-regulatory circuitries? Okay, so I will try to explain. CRC, core circuitry, is a set of transcription factors with certain properties. So each transcription factor is encoded by a gene with a super enhancer, and there are motifs for the product of this transcription factor in the super enhancer. So you have positive feedback loop. So each transcription factor binds to its own super enhancer and usually activates, most of the cases, its own transcription. And all transcription factors in this set of the CRC have these properties. Moreover, they have binding motifs for all transcription factors in this list. So they all bind to each other's super enhancers and activate each other. So it's a regulatory click, if you want, in the regulatory graph. And to find them, people just locate super enhancers, do motif analysis, create these graphs, and find clicks in the graph. To give you an example of such CRC, so this is a cochlear circuitry identified in human embryonic stem cells. So about 10 transcription factors, all the super enhancers, all activate each other, and they can also bind to super enhancers of other transcription factors and activate them, but there is no this positive feedback loop from other transcription factors to this core. And in this core, if you look closer, you will see OCT4, SOX2, and NANOG, and you must know these transcription factors, right? Nobel Prize. So if you ectopically activate these 3 transcription factors plus 1, I think LINT1B or MYC, you can reprogram fibroblasts into induced pluripotent stem cells. So the fact that you see this core CRC, including OCT4, SOX2, and NANOG, by doing this analysis shows that the method probably works. So we wanted to identify this CRCs in neuroblastoma and see how it changes. But before going to biology part, I wanted to talk about methods that we had to develop to better analyze the ChIP-seq data. So there are 3 parts in this method section I'm going to talk about. Yeah. It's a good question. I have an intuition about this. So the question was, what is a cell identity? Difficult question for me. So,
but people use it in these papers, yes? That's...
Yeah. So practically, for me, cell identity is the different transcriptional profiles, which is translated in different phenotype, like high migration potential because you have podia and the cell can move, or higher proliferative rate. But generally, when I speak about identity, it's in the context of differentiation, when you have stem cells that divides and changes identity during the differentiation, so you get muscle cells, or nerve cells, or neurons, and so on. All right, so the first thing what you do when you do analysis of ChIP-seq data is peak calling, which is to go from reads that you sequence to regions, to density calculation, and then identification of regions with a signal. So I just forgot to mention to you what is ChIP-seq, right? So I think I will have to explain. So ChIP-seq is a technique to obtain profiles of histone modifications or transcription factor binding sites. So the purpose is to see where a given protein binds to DNA. And this protein can be transcription factor, RNA-POL2, or a histone, which is modified on some lysine, for example. And the way to obtain this information about where the protein binds to DNA, you first cross-link the protein to DNA, so the protein cannot unbind, so it's fixed to DNA. Then you shear DNA into pieces, usually with ultrasound. And then you take an antibody, which is specific for your protein of interest. You immunoprecipitate DNA protein complexes that contain your protein of interest. Then you reverse cross-links, you purify DNA, and in the end you get a library of DNA fragments each of these fragments was bound by a protein at some point. So you try to enrich in DNA fragments where the protein binds and remove everything where protein doesn't bind. And then you usually do size selection to get fragments of certain length, for example, 300 base pairs, and you sequence one end or two ends of these DNA fragments. You map them to the reference genome, and if you see an enrichment in mapping reads, so these sequence fragments, you can think that the binding was taking place in this region. But this mapping is difficult to read, so you want to analyze it, you want to go from these reads to densities. And usually if you read a paper about a ChIP-seq analysis, you see rather this density plots. So how do you go from reads to densities? By peak calling, and there are many methods to do it. The most highly used are CISR and MAX for histone modifications, but they have been developed for diploid genomes, and here I wanted to show that they don't really work well on cancer genomes because they have a huge copy number bias. So here I'm showing to you a piece of chromosome 8 that has a loss on the 8p region and again in the 8q region. And this is the output of CISR and MAX. Each small rectangle, it's a region called by these tools. And you see that in the region of loss, these two tools didn't really call lots of regions. So this is binding of K27 trimethylation. So we wanted to create our own method, which will not have this copy number bias. And we did it in 2013. We called it HM-CAN for histone modifications and cancer, published in Bioinformatics. And in addition to this copy number bias, HM-CAN corrects for GC content bias and library size bias. And then on these normalized densities, it applies hidden Markov models to segment the genome into regions of signal and no signal. And it turns out that having just two states in this hidden Markov model is enough to have very good predictions. And to get these copy number aberrations, we use the input sample that is present in every ChIP-seq experiment. So I forgot to tell you that when you do ChIP-seq, so one experiment when you use an antibody, and you have a control experiment, which is called input, where you just cut DNA into pieces and sequence. And this input experiment is actually low coverage whole genome sequencing. And you can get lots of information from it, including copy number, and actually contamination of your sample by normal cells. This is what we also do. So here I'm showing you the copy number profile that you can obtain from ChIP-seq data for HeLa cells. And just if you work, for example, with ATAC-seq, you don't have this input control, but our method can actually extract this copy number information from ATAC-seq as well, just by masking the regions of enrichment, looking at the background regions and constructing these copy number profiles from it, and it works really well. So actually you don't need the control experiment anymore. All right, so as you can see here, the output of HM-CAN doesn't have this copy number bias, so in the region of genomic loss you still have the signal, which is good. All right, so this was the first step for my project, identify regions of enrichment. The second step is to identify super enhancers. And as I told you, the canonical method to find super enhancers is called rows, and it's based on simply counting number of reads for K27 acylation minus the number of reads in the input control and ranking candidate super enhancers according to this number of reads. But imagine in cancer, you are in a region that was amplified 100 times. So you will have 100 times more signal for acylation, but also in the control experiment. If you do just this difference here, even if you have a small enhancer, it will get a very high score, and you can call a small enhancer a super enhancer if you allocate it, or if this region is located in the amplification region. So we wanted to modify this method, and we called it the new method Lily. And it works exactly the same, but it doesn't use read counts, but it uses normalized density profiles constructed by the previous method HM-CAN. So you normalize your read counts by GC content bias and copy number bias, and only then you do this calculation of sums of densities, you do your ranking and so on. And this allows you to identify super-enhancers in the region of genomic loss when you have one copy left instead of the two. And also it allows you to avoid false predictions of super-enhancers in the regions of genomic amplification. And I'm showing you an example here. So these are this is ChIP6 signal for K27 acetylation in 4 different neuroblastoma cell lines. This first cell line doesn't have an amplification in the MECAN locus, so you have 2 copies. And these 3 cell lines, they have an amplification, so you have more than 100 copies of the MECAN locus. And the signal I'm showing to you is a signal after the normalization by HMCAN. So you see that the profiles look very comparable, so you removed all copy number bias. You can clearly see activated promoter for MECAN, which is here, lots of enhancers. And if you apply Lilly on these profiles, it will predict the super enhancers in these 3 cases, and it will say that here is just a bunch of enhancers and it's not a super enhancer. All right, so this was the second step for the methods, super enhancer prediction. But then imagine you identified the super enhancers and you want to know which transcription factors can drive these super enhancers. For this, we usually do motif discovery analysis. We look for transcription factors motifs which are enriched in these enhancers or super enhancers. But our super enhancers are very large. So here it's a super enhancer which comprises almost 500 kilobases. It's huge. And you're looking for enrichment of motifs which are very small, like GATA motif, 5 letters. How can you find an enrichment of something which is very small and region which is huge like this? Only if the signal is very strong. So what we suggested is that you should not look for motif enrichment in these regions, but instead you should zoom in to peaks here, find valleys in the signal in these peaks, and look for motifs in these valleys, because these valleys correspond to putative transcription factor binding. So this is experimental data for some transcription factor. This is a K27 acetylation profile. And why you have these valleys? This is because here you measure signal on the nucleosomes, right? And if transcription factor binds, there is no nucleosomes in this region, usually. So it's an open chromatin region. So when you have a binding of transcription factor, you will have a gap in K27 acetylation. So in our package Lilly, you can find a tool that will take your super enhancer regions, this density profile, and it will find these valleys for you so that we can do motif enrichment analysis only in these valleys. So all your p-value will be much more significant. All right, so this is the end of the methods part. So the most important conclusions are that you have to explicitly correct for copy number when you deal with the ChIP6 data coming from cancer cells. Yeah. So we don't use p-values to find valleys. I was maybe too lazy to write a very sophisticated method to do it, so I just looked at the profiles and I look for regions where the gap is deep enough compared to neighboring regions. No, computationally, but I don't use any statistics to do it, just empirical things like it should be twice lower than neighboring. But if you want to write a good method, you can. No, so here it's not a statistical method. All right, so now I can speak about neuroblastoma and a couple of words what this cancer represents. So neuroblastoma, it's a pediatric cancer. Average age diagnosis is 18 months. So exactly, it's not a brain cancer. It's always extracranial. It can be in head and neck, but it's always outside of the brain. And the most common location is adrenal glands. Good. So since it's a pediatric cancer, you have very few genetic events. The most common genetic event is amplification of the MEK and oncogene, found in about 1/4 of patients. And the second common event is activating mutation of the ALK oncogene in about 10% of patients. Hereditary neuroblastomas are very rare, and in these cases, we sometimes see the ALK mutation and deletion or mutation in the FOX2B transcription factor. So we wanted to profile super enhancers. For this, we generated ChIP-seq data for K27 acetylation in 25 neuroblastoma cell lines and 6 patient-derived xenografts to show that what we see in cell lines makes sense and it's similar to tumors. And as a control, we used neural crest cells because neuroblastoma is believed to derive from neural crest. However, we don't know at which stage neuroblastoma derives from neural crest. And I think there are methods to actually see it by looking where mutations are located and comparing the different landscapes. But this work has not been done. So far we don't know at which point of neural crest development neuroblastoma can develop. And in addition to K27 acetylation data, we also had RNA-seq data to correlate the activity of enhancers and super enhancers to the gene expression. So all experimental work for this project was done by the team of Isabelle Janouex from the Curie Institute, and my team did bioinformatics. I'm going to show you 2 examples of super enhancers we have identified. The first one was in the ALK gene. So I remind you that ALK is the most commonly mutated gene in neuroblastoma. And if a patient has a mutation in the ALK gene, then he will be treated by the ALK inhibitors, crizotinib or others, and it really works. However, what we show is that, at least in cell lines, you can have a super enhancer in the ALK gene, even if ALK is not mutated. So maybe patients who have a super enhancer in ALK, but ALK is not mutated, can also benefit from ALK inhibitors, and this is what we try to check now in collaboration with a group from Charité, Berlin. So here I show you K27 acetylation signal for 4 different cell lines of neuroblastoma in red and 2 normal controls in blue. And here you have cell lines which are ALK mutated or ALK wild type, MEK amplified, and cell lines where you don't have amplification of the MEK gene. And you see that the profile is very similar. And the region of the super enhancer is very long, so it's about 500 kb. Another example is for FOX2B gene. So I told you that mutation in FOX2B can predispose to neuroblastoma, and these are deleterious mutations. And it was quite strange for us to see the strongest super enhancer in the FOX2B gene. So here again you have these 4 cell lines, and you have this strong K27 acetylation signal for cell lines where you have ALK wild type, mutated, MEK amplified or non-amplified. So here I just showed you 2 examples of super enhancers. Yeah.
So when you say a super enhancer in a gene, isn't enhancer something similar to promoter, which is like next to the gene, and like is it like far away, or like why is it on the gene?
So the question is, how do you annotate super enhancers? How do you assign super enhancers to genes? It can be located. It can overlap the gene, like here. You can see that this super enhancer actually overlaps this gene too, but I don't assign the super enhancers to this gene, right? Here it's in the gene, so sometimes it's close to a gene. So how I assign super enhancers to genes? First, I take all available data for topologically associating domains. So these are these loops of chromatin. And I assign a super enhancer to all genes in the same topologically associated domain. It can be assigned to 10 different genes. And then I look at the correlation between the strength of super enhancer, which is just the sum of densities in a given region, with gene expression for every gene in the same TAT, with the FOX2B, with this gene here, and so on. And I have a threshold which corresponds to 5% of the p-value, I'm sorry, to say that this super enhancer activates this gene. And sometimes it's assigned to one gene, sometimes it can be assigned to 2 genes, and I think it's biological reality. I think one big super enhancer can actually drive expression of several genes in the region. All right, so you cannot compare super enhancers one by one, cell lines by one. Instead, you can do dimensional reduction techniques, in this case PCA, to see whether a super enhancer landscape of one cell line looks similar to a super enhancer landscape of the other cell line. So each point here, it's a cell line. The color code shows you the genomic aberrations. For example, green ones are MECAN amplified cell lines. PDXs are shown by triangles, and 2 normal neural crest controls are shown in blue. So you can see that normal controls, they're quite apart from the majority of the neuroblastoma cell lines, but you have 3 cell lines for which the super enhancer landscape is very similar to normal neural crest controls. So we define group 1, common group, and this group 2 that they call neural crest-like cell lines. And if you look at the strongest super enhancers and genes associated with super enhancers in these 2 groups, we see FOX2B for group 1, ALK, TBX2 that has a very strong super enhancer in both groups. And if you look where this TBX2 gene is located, you will see that it's located in a minimum gain region on chromosome 17, which is gained in more than 80% of high-risk neuroblastomas. So you can think that this gain on 17q is one of the first events in neuroblastoma to amplify genes in this region, including TBX2, and there's high expression of TBX2 can start with this rewiring by binding to its own promoter, creating super enhancer, and so on. And the color code here corresponds to the fact whether a given super enhancer has been already seen in the neural crest cells. So blue means that the same super enhancer is present in neural crest, and orange means that de novo compared to neural crest. So you see that group 2, these are cell lines which on PCA were located close to neural crest. They're all blue, and here they're all orange. So we wanted to know what drives the difference between these 2 groups, and we looked which genes or which super enhancers contribute the most to these principal components, and we identified FOX2B. And when we did a Western blot for the FOX2B protein, we saw that indeed in these 3 cell lines here, FOX2B protein was absent, and it was also absent in SK and SH cell line, which is also very close to neural crest on the PCA. So they thought that maybe FOX2B is the president that drives the identity of these cells, but we wanted to find a president of this group and also ministers and so on. So we applied motif discovery to see which transcription factor motifs are enriched in super enhancers of group 1 and group 2. For group 1, the strongest hit was this motif, which looks like FOX2B motif. So we confirmed that FOX2B is an important transcription factor driving the identity of super enhancers, driving super enhancers in group 1. And for the group 2, we saw very strong enrichment in AP1 motif. So AP1 is a complex of FOS and JUN transcription factors. It can be any transcription factor from this FOS and JUN family. It can be FOS, FOSL1, FOSL2, JUN, JUNB, JUNZ, and so on. It's always the same motif. So we thought that we identified 2 presidents for these 2 groups, but we wanted to know more what are other transcription drivers. And then we applied the method developed by the group of Rick Young to find these CRC networks that include FOX2B, FOSL1, JUN, and so on. And here I'm showing to you transcription factors that are included in the network of group 1. So this is group 1 cell lines and transcription factors that are included in the CRC networks of group 2, which is this one. Sorry, you can see that these transcription factors are also in the network of these 2 normal controls. So we thought that maybe these are the other players which are very important for the cell line density of group 2 and group 1, but we wanted to narrow down the lists, and the way to narrow down is to look at the correlation of gene expression, because if you are involved in a very dense regulatory network, so for example, I activate Eva, Eva activates Ben, Ben activates me, our expressions should be correlated, right? So we looked at the correlation of gene expression for these transcription factors here and there, and we found groups of very high correlation. So one group including FOX2B, FOX2A, HAND1, HAND2, GATA3, and the other one including FOSL1, FOSL2, PRX1, RUNX1, and so on. And this is the corresponding correlation graph. So you see that on the cell lines, these transcription factors are correlated, the expression is correlated, and the expression of these transcription factors is correlated, that they are anti-correlated in between. So we maybe think that this is model 1, this is model 2, but these correlations were obtained on our cell lines. So you cannot publish something only on cell lines, so you need to go to tumors. And fortunately, we had a data set for 500 neuroblastoma tumors, and we correlated correlations on these tumors. And it was not as black and white. So for module 2, we see a very high correlations, but for model 1, it was not as obvious. So we narrowed down our model 1. So we say model 1 is FOX2B, GATA3, and HAND2, and these genes are anti-correlated to these genes. So we propose that there are 2 modules that are, it's either one model which is on or the other. And we validated by ChIP-seq the first module. So here I'm showing to you ChIP-seq data for HAND2, FOX2B, and GATA3, and K27 acylation in the FOX2B super enhancer. And here you can see that there is not just one binding site or 2 binding sites. You see that there are 20 different binding sites for these transcription factors. They always bind the same region in the valley of K27 acylation. And so this is super enhancer FOX2B, so you have this auto-regulatory loop. All right. So if you believe that we have identified this model 1, model 2, then you can characterize a given cell line or a given tumor by the expression of transcription factors of model 1 and model 2. And for this, we simply did average gene expression in log scale of these 2 groups. And we plotted it for neuroblastoma primary tumors. So on the x-axis, you see average expression of these 3 genes, FOX2B, GATA3, and HAND2 from model 1. And on the y-axis, you see the expression of transcription factors from module 2, PRX1, FOSL1, and so on. And each triangle is a tumor. Median amplified tumors are shown in blue. And we were quite disappointed when we saw this picture because we were expecting to see 2 clusters. One cluster expressing model 1 and one cluster expressing model 2. And it was not the case. So after thinking for some time, we thought maybe tumors are heterogeneous. Maybe in the tumors, we can have both types, both identities. And we did a single cell expression analysis. So in our paper, we applied biomark technique on this cell line SKNAS. And we show, so here you have data for 48 cells, and you see that there are cells from this intermediate cell line that express model 1 genes, and there are cells that express model 2 genes. It's not very clear, so we repeated this experiment by applying the 10x single-cell RNA-seq technique, and this data are unpublished. I'm very sorry for the resolution. So you almost don't see the gray clusters. So what I'm showing here is single-cell RNA-seq data for SKNSH, mixed cell line, and this is the expression of FOX2B, PRX1, and FOSL1 in the t-SNE dimension reduction space. And you have here 3 clusters. One cluster here expressing FOX2B, and 2 clusters here expressing PRX1 and FOSL1 and the group 2. So we confirm that cells that have the same genetic background, the same mutations, copy number aberrations, they can be in 2 very different transcriptional states that are governed by 2 different sets of transcription factors. We showed it on cell lines, and our competitors rather, right? Our competitors showed it on tumors, and for this we applied imaging techniques. Here you see a piece of tumor where all neuroblastoma cells are shown in blue, and neuroblastoma cells expressing PRX1, which is a part of module 2, it's shown in yellow. So module 2 cells are yellow, module 1 cells are blue, and you see that you have both of them in the same tumor. Moreover, you don't see a cluster of yellow cells or blue cells. It's all like salt and pepper pattern. It may mean 2 things. Either there is some plasticity, so a yellow cell can become a blue cell, and blue cell can become a yellow cell, or they need each other. A blue cell wants to be in a contact with the yellow cell. And we just got money to explore more on this. I will skip this. And what was also interesting is that cells with different phenotypes have different sensitivity to chemotherapy. So to show it, we took this mixed SKNSH cell line that has 2 types of cells, module 1 cells, module 2 cells, and we applied 2 different chemotherapeutic treatments, cisplatin and doxorubicin, and we measured the expression of module 1 genes and module 2 genes. And we saw that after treatment, the expression of module 1 genes goes down, and the expression of module 2 genes goes up. So it may mean 2 things. Either module 1 cells die as a result of chemotherapy, or they reprogram into type 2. So far, they think that both things happen. Mostly they die, but some of them can actually reprogram to escape the chemotherapy. So to conclude, I would like to repeat that in neuroblastoma, we think that cells can be in 2 different states. One state, which is more common, and it's actually the major state for MECAN amplified tumors, is governed by transcription factors FOX2B, GATA3, and HEN2. The other state that we call neural crest-like state or mesenchymal state is governed by FOSL1, FOSL2, PRX1, and so on. These 2 cell states can be present in the same cell line or in the same tumor, and module 1 state cells are much more sensitive to chemotherapy. So maybe when a patient is diagnosed with neuroblastoma, we should look at the percentage of module 2 cells and maybe increase chemo or combine chemo with some other treatment that can reprogram module 2 cells into module 1 cells. On this, I would like to thank people who participated in this work. Biologists from the Azerbaijan UX group, Caroline, Simon, Agath, bioinformaticians from Curie with whom I started the work on this project, Irina Medvedeva, former postdoc in my group who helped me with the analysis of ChIP-seq data, and Haitham Asher, former PhD student who developed HMCAN and other techniques to analyze ChIP-seq data. I would like to mention that we have an open postdoc position to continue on this project in my team, and thank you for your attention.
Thanks, that was great. So for the module 1 and module 2 type cells, can you say something about the rest of the expression program? Like, does one of them look like it's maybe cycling more, or does one of them look more or less differentiated, or is there anything else you can tell?
Yeah, so you can think about neural crest-like cells, module 2, as less differentiated, For proliferation and cycling, they cycle the same, like very, very aggressive, both of them. If you graft the cell lines into a mouse, both of them will grow very fast. You have differences in transcriptional programs for sure. Some oncogenes are turned on in one cell type and other oncogenes are turned on in other cell type. So I think B-core is in module 2, or MYC-N is under the control of FOX2B, HEN2, and GATA3. So the programs are very different.
So it seemed like in Module 1, FOX2B is part of the core regulatory circuitry and also mutated. So is that considered an initiating event for that reprogramming? And is there a similar initiating event for type 2?
It's very interesting. So FOX2B is present as a major, like a president of group 1, but it can be mutated in hereditary neuroblastomas. So my explanation is that if you have it mutated, then you're likely to develop this group 2 neuroblastoma, so this more maybe aggressive type.
So it's sort of a switch. If you mutate, you switch types. So it's not then thought to be the initiating event for
it can be one of the initiating events, but if you have it mutated, you will not have model 1, right? You will have model 2 active.
Good. Any other questions? All right, if not, let's thank Valentina and we'll move on.